Hello, my name is Kyle Malter. I'm currently working with the Shikuma Lab, and we're trying to answer the question whether bacteria can be used for therapeutics. So we study a marine tube worm and how bacteria interact with it. Here's a small bacteria. And this bacteria is necessary for this marine tube worm to undergo a metamorphic change from its larval state into its juvenile state. And without this bacteria, this tube worm would stay in its larval state and would never become an adult and reproduce. So these bacteria produce a unique structure that is virus-like. It sort of looks like a virus tail. And there's a small tube that injects almost like a needle protein into these tube worms. So what we're trying to determine is the signal sequence necessary for this protein to be injected into our tube worm. So here's our tube worm. And this protein is injected into our tube worm, and that's what's causing this metamorphic change. Now, these structures are also specific. So they have these tail pins that bind specifically to our tube worms, and if these tail pins are missing, this interaction cannot occur. So that allows us to determine the specificity of these structures to our tube worms. And so by determining these two things, we can use this structure potentially to load a specific protein using a signal sequence and target these structures with the tail pins using its specificity to human cell lines like cancer cells and target them for degradation or apoptosis. Or we could use them for beneficial things like targeting them to pancreatic beta cells to induce proliferation or reproduce. And so by determining the specificity of the structures and the signal sequence needed for them to load proteins in these structures, we can answer the question of whether bacteria can be used for therapeutics. Thank you.